Hello, everyone. If you're new to our webinar series, welcome and thank you for joining us. Criterion Edge runs multiple informative webinars throughout the year. For today's webinar topic, we are discussing leveraging clinical data in the Clinical Performance Report. Our presenter today is Dr. Sarah Chavez, Principal Medical Writer with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. Sarah has extensive scientific writing experience in an academic environment as well as for several large textbook publishers. Her strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. And now I'll welcome Sarah to give an overview of our session today.
Okay, thank you, Olivia. So as a disclaimer, today's session is being presented from the point of view of our experience as seasoned MDR-compliant CER medical writers. So we will be discussing IVD in the context of our previous experience writing for MDR, but we hope that in today's presentation, it will help you identify meaningful and measurable performance measures, and also to address sources of data to support the Clinical Performance Report, or CPR. So the takeaways for this presentation will be to discuss the purpose of the CPR and its role as a component of the PER, to identify sources of clinical data and to analyze those data within the context of the performance endpoints and the acceptance criteria that we've discussed and that have been previously presented in the SVR, and then to demonstrate how to extract, organize, and appraise these clinical data within your CPR. The PER ultimately has its roots in the Clinical Evaluation Report, or the CER, that's required under Medical Device Regulation, or MDR. The PER is really the output for the performance evaluation process, and it uses the results of your performance evaluation plan. If you refer to Annex 8 within IVDR regulations, that's where it really outlines the specific components required within the PER and describes that it must include your scientific validity report, which we've spoken about previously, the analytical performance report, and the clinical performance report. However, these reports provide the necessary clinical evidence, but IVDR also requires an assessment of those reports that supports your assertion that the clinical evidence is sufficient to make a decision on the benefit-risk ratio. So your PER should also be ready to describe the methods for gathering clinical evidence. That will include things like literature searches and their related protocols and reports, a description of the technology behind the IVD, your intended purpose and associated claims, the actual scientific validity, and the analytical and clinical performance data that are under evaluation, and ultimately the clinical evidence that supports the use of this device when assessed in the context of the current state of the art. So for many companies, you may have prior experience with the FDA 510K submissions for your devices. So you may actually already have some of the elements for the PER in place. However, the PER must support the clinical benefit of the IVD test, which is a new requirement. And there will most likely be additional data that will need to be generated and gathered as appropriate. So first off, what are the types of clinical evidence? These definitions should be used to help guide how and where the clinical evidence will appear and be presented within your PER. So we'll start with just a very brief recap of the definitions for each report. The scientific validity report should provide evidence supporting the association of the analyte with a clinical condition or a physiological state, which is quite similar to the purpose of the SOA section within a typical CER. The analytical performance report will present data relevant to the ability of the IVD to correctly detect or measure a particular analyte accurately, as opposed to the clinical performance report, which is focused on the ability of that IVD to yield results that are correlated with a clinical condition in a specific target population under normal conditions of use. In the first part of our deep dive series, we discussed strategies for PER planning and described the importance of making some of these decisions very early, particularly with regards to your clinical performance objectives. So in that webinar, I used the example of COVID testing to explain how to extract safety and performance objectives and how to use these data to derive the acceptance criteria. I will reference many of these same concepts in today's deep dive part 2, but I will be using another real world example to hopefully help explain the purpose of the SVR and to distinguish between the alternatives and the competitors in your analysis. Now, suppose in this instance, this example, your IVD is an immunohistochemistry test for a breast cancer biomarker called HER2. Your first task is to establish that HER2 is associated with breast cancer and to explain how this association is clinically relevant. HER2 is a human epidermal growth factor receptor 2. And previous studies have shown that HER2 is overexpressed in 15 to 20% of breast cancers, and it happens to be associated with a much more aggressive disease course. So in order to describe that association, you have to establish the who, what, where, when, and why of HER2 testing. Why is it relevant to clinical diagnosis and treatment decisions? So for instance, current guidelines recommend HER2 testing on all newly diagnosed patients with invasive breast cancers. So in the SVR, you should be prepared to explain how knowing whether a tumor expresses this analyte affects clinical decision-making. And again, in this example, HER2 can be used for diagnosis, but it can also act as a companion diagnostic tool to help with treatment decisions. Some breast cancer treatments are approved for HER2 positive disease, while others are approved for HER2 negative tumors. Next, your SVR should describe alternative ways to detect HER2, and those are presented here in the table. Your IVD may be an immunohistochemistry test, so those will be your competitors, our other IHC tests. Alternative diagnostic testing options may include technologies such as FISH or a PCR type of technology. And what about sample types? Can your test be used on plasma in a liquid biopsy, or do you need tissue samples? Does that tissue have to be fresh or frozen? All of this information speaks to the association of the analyte with the clinical condition and supports your scientific validity. But really, your goal for the SVR section is to show that your IVD may be considered state of the art with regards to the ability of that test to make this diagnosis. So how do you define success? Are you using it to diagnose a clinical condition? In order to show that your IVD performs as intended, you must show that it has sufficient sensitivity and specificity compared to your nearest competitors. That's how you will ultimately set the bar. And that's the purpose of the SVR that we described in the previous webinars. It's to establish your competitive landscape by showing how your closest competitors perform. Now, our first deep dive, we explained how to extract data for safety and performance and how those data can be used to actually derive these acceptance criteria. The SVR will show how well your alternative testing methodologies perform, but it will also present data from relevant clinical practice guidelines. In this case, the acceptance criteria you've established is based on how well your competitors perform. So if your competition has a sensitivity of 96 to 98%, as you can see in the highlighted column there, your acceptance criteria should be at least 96%. Now, you would not want to compare your device, which is an IHC test, to a PCR test. That has a much higher sensitivity. So really, this is the purpose of that competitive landscape that we've written in the SVR. So you can establish the criteria by which you will be judging your own test's performance. Are you at least as sensitive as your competitors? Do you have fewer false positives or false negatives than they do? The ability of that test to measure that analyte will be discussed in the APR. But while your test may be able to accurately diagnose 100% of samples over a specific limit of detection, that test could still have some limitations in a real world setting. And that's why the CPR is here. And that's why we're being tasked with collecting both analytical and clinical performance data. We can use the performance of HER2 tests in real world clinical settings to justify the intended purpose of that test, and ultimately to support the clinical benefit of testing. So while the scientific validity report will justify the approach that you took in terms of how you're detecting it, it will also provide evidence supporting that that analyte is associated with more aggressive disease. So in effect, the scientific validity report is providing the support and the justification for the clinical performance data that you will be extracting in your CPR. So that brings us to today's webinar. Now that you've established your acceptance criteria using data from your competitors, how do you show how your IVD performs by comparison? Up until now, it's important to keep in mind that that clinical evidence that you've presented has all been on the safety and performance of your alternatives and competitors within the state-of-the-art section. So using the acceptance criteria in the SVR, the PER must now present the data pertaining to the analytical and clinical performance of the subject IVD in the APR and the CPR. So first, I think it's helpful to clarify the conceptual difference between analytical data and clinical performance data. Just because an experiment uses patient samples does not make it a clinical data set. So first and foremost, analytical performance and clinical performance studies, they have different objectives and endpoints. So really the first clue as to which type of report your data will be featured in is the type of data that you're extracting. You can think of it like this. An analytical performance study is focused on that analyte, whereas the clinical performance study is focused on the patient. So yes, they are inherently related because analytical performance is the basis of that clinical performance. But analytical performance data do not directly demonstrate the clinical performance of a device, as they are fundamentally different performance characteristics. Your test may perform with precision, accuracy, and reproducibility in a controlled laboratory setting. But when you bring it into a real world clinical scenario, how well does that test perform? Are there situations or patients in which that test may not perform as well? And the answer is perhaps. What if your patient is already receiving treatment? Does that affect the levels of HER2 that are detectable? Are there characteristics of the tissue or other reasons why some patients may be more likely to receive a false positive or a false negative result? To give you a real world example of this, the 2013 guidelines state that when you're making a diagnosis for being HER2 positive using FISH, you should include the average number of HER2 genes per tumor cell, as well as the ratio to an internal control gene. Now, that assessment creates 5 possible groupings, and one of those groupings is classified as HER2 equivocal, meaning that it's not higher and it's not lower. It's sort of indeterminate. The 2018 update tried to clarify this by describing which types of internal control genes should be used and to warn which type actually may lead to false positives. And in fact, a later study assessed the use of recommended control genomes in order to assess HER2 positivity. Now, these tests performed precisely as intended in an analytical sense. So their analytical performance data may be entirely accurate and reproducible and precise. But in a real world clinical setting, some of these controls resulted in substantially higher rates of false positives than others. So that's why the real world data matters and why IVDR is making this requirement for clinical performance data within the CPR. IVDR gives us a few hints as to what they expect out of these clinical performance studies. So first, IVDR Article 57.2 states that where appropriate, a performance study shall be performed in circumstances similar to the normal conditions of use of the device. And it goes on further to state that the characteristics and performances of the device shall be specifically checked in the event that they may be affected when the device is used for the intended use under normal conditions. That gets into that real world scenarios, anything that can result in unexpected results, even if the device performs as it was originally designed. And finally, clinical performance studies need to be designed in such a way as to maximize the relevance of the data while minimizing the chance for potential bias. So these definitions are fairly straightforward, but they really don't provide us much direction in terms of where to look for all of this data. And really, that's been a particular concern for many of our IVDR clients. What do we do if we don't have any clinical data? So our first step is to assess whether or not this is actually a true statement by searching across multiple sources of potential clinical performance data. For some companies, you may have manufacturer-held data, which is an excellent place to start. There are also these external data sources, which are basically anything that occurs outside of the walls of your facility. These can include clinical registries and public data sets that may include published clinical data pertaining to your particular IVD. And finally, your best hope may be to rely on the kindness of strangers who have used your device in their own studies and were nice enough to publish that data for you. So that's why systematic literature review is required. It's your opportunity to thoroughly and objectively search across published articles in multiple platforms and across multiple databases. So data sufficiency is a concern, but a well-designed systematic literature review and looking at all of these different potential sources of clinical data can ensure that if the information is out there, you will find it. As you look through this list that I have here on the right-hand side, you can see that it would be you wouldn't necessarily want to put the same weight on a case series that includes 5 patients as you would a meta-analysis with potentially thousands of tests that were run. So this is really a good time to point out that when you're presenting these data sets, it's critically important to also provide weighting and appraisal of your data. This is ultimately what the appraisal criteria, this is similar to the appraisal criteria that we would use in a typical SVR or an SOA section. For each of the identified clinical data sets, they should be appraised for their suitability and data contribution towards the clinical evaluation. And I have the regulation listed there at the bottom. For each identified data set, the methodological quality and scientific validity will be evaluated, and the relevance of the information will be determined for each independent data set. And in that case, it's each individual study. So the data should be intended to directly demonstrate adequate clinical performance and clinical safety. And you can also include data that's serving more of an indirect supporting role. So some of this non-pivotal data may be helping you to identify current knowledge or state of the art, or potentially for the identification of hazards or risks. So yes, those case studies are not necessarily weighted the same as you would a meta-analysis, but they can be incredibly important for the overall report, particularly if they've identified a potential cross-reactivity or a hazard or a risk associated with your test. So in this example, we have several different levels that you can see here. So when you have a level one is data on your subject device when it's used as intended in the appropriate patient group where the study designs are of high quality. So it's basically your top tier data. That is the data that's most directly relevant and pertaining to the clinical performance of your subject device. But it's also recognized that it's important for manufacturers to understand off-label usage of the subject device in patient groups. So that's where the level 2 data comes in. They may have minor deficiencies in terms of the data collection or the data quality, or they may be used for an off-label purpose, but this data should still be captured and weighted and appraised as a level 2. And then finally, the level 3 data. That's the data from the current knowledge and state of the art and from similar competitor IVDs for both on-label and off-label use in all patient groups. And that, again, may have high quality or minor deficiencies and includes multiple different study designs, but they should reflect the performance, an appropriate follow-up period, appropriate statistical analysis, and ultimately a clinically significant effect. So if you have reports that have met these criteria, they will be critiqued and analyzed. Each one of these articles will be analyzed as part of your performance evaluation. The analysis will determine whether any potential risks associated with the intended purpose have been minimized and are acceptable when weighted against the benefits to the patient. And that's how you will support the safety and performance of your subject IVD. So that finally brings us to the role of the clinical data in the PER. Now that you've established your acceptance criteria and you've shown how your IVD performs by comparison using clinical performance data, how do you use that data to support clinical benefit? In IVDR Annex 8, they state that the manufacturer shall assess all relevant scientific validity, analytical, and clinical performance data to verify conformity of the device with the general safety and performance requirements. And the amount and quality of that data, and quality gets back to that appraisal, shall allow that manufacturer to make a qualified assessment as to whether that device will achieve the intended clinical benefit when used as intended. So based on this definition, the manufacturers must describe the intended clinical benefit based on your intended purpose statement, and then perform a qualified assessment of the acceptability of the benefit and risk of that device using the clinical evidence to assess whether or not a clinical benefit was actually achieved. That can be a qualitative assessment based on the judgment of the person taking into account the diagnostic information on a patient as provided by the state of the art. The intended clinical benefit should be described in the performance evaluation plan, while the assessment of the benefit risk and the presentation of that clinical evidence supporting the benefit should be clearly documented within the performance evaluation report. So that's the main difference between the PEP and the PER. The PEP is stating how you are planning to collect this data, and the report is ultimately what did you find when that plan was put into place. And again, kind of referring back to our original definition and clearly defining what is a clinical benefit. Is according to MedDev Rev 4, the clinical benefit can be defined as the positive impact of a device on the health of an individual expressed in terms of a meaningful, measurable, patient-relevant clinical outcome, including outcomes related to diagnosis, patient management, or public health. Now, while direct clinical benefits should be supported by clinical data, there could also be indirect clinical benefits that may be demonstratable by other evidence, such as preclinical or bench test data, real-world data sets, such as those registries, or information derived from insurance database records, as well as data from another device that's used with the subject device that may not have direct clinical data. But I'd like to just point out a key distinction between clinical benefit in terms of how MDR and IVDR define it. Unlike medical devices, which have a direct impact on patient outcomes, the clinical benefit of an IVD is the ability to provide accurate clinical medical information, not the final clinical outcome for the patient. So in conclusion, we'd like to remind you of what the notified body expects to see in the process of presenting your data within the CPR. They expect that you will have identified meaningful performance endpoints, but also support the appropriateness and the clinical relevance of these performance endpoints in the context of what can be considered state of the art. They want to see an indicative list and specification of the parameters that you use to establish your acceptance criteria. And then finally, they expect to see actual analytical data and clinical data pertaining to your IVD. And that will be used to determine the acceptability of the benefit risk ratio for the subject IVD. So we hope that in today's webinar, we've helped to illustrate the purpose of that CPR and its role as a very critical component of the PER, to provide examples of the sources of clinical data and how to analyze those data within the CPR in the context of the performance endpoints and the acceptance criteria that you've already presented within the SVR. And then finally, to demonstrate the importance of knowing how to properly extract, organize, and appraise these clinical data sets within the PER.
Thank you, Sarah. That was a great presentation, some wonderful information there. We will now turn to our question and answer portion of our presentation today. So if you have any questions on any of the content Sarah presented today, please go ahead and use the Q&A button on your Zoom menu bar and send us your questions. We have about 30 minutes now to answer your questions. So now let's get to our first question. Sarah, let me know when you can see it on your screen.
Okay, yes.
Okay, today our first question is, how is clinical data and clinical evaluation report relevant to IVDR requirements?
So I think this is really getting at the question of where do we find information and guidance, because there really is so little IVDR guidance available to us. So really, when we go to MDR, that seems to be what the majority of notified bodies are referring us to, is to go back to the guidance for MDR. And we know that the IVDR guidance is forthcoming. It's supposedly on its way, but we also know that there's going to be a lot of people making these submissions in a very short amount of time. So your time constraints could be very, very limited. So ultimately, I would say, in terms of clinical data and the relevance, if you have a doubt about how to present information for IVDR, see what the equivalent guidance is for MDR while we're waiting for that guidance to come out. It's on its way, but for now, the majority of people are referring us to MDR guidance.
All right, thanks, Sarah. Here's our next question. How exacting are the criteria for proving equivalence?
So these are clearly going to be a major issue for a lot of our, establishing equivalence is sort of that a point of confusion for a lot of our clients. So it could be in terms of technical equivalence. That would be where you would describe that your particular test is of a similar design or used under similar conditions with similar specifications and properties. It could be a biological equivalence, meaning that you're using the same materials or substances for a similar kind or duration of time, or that it has similar degradation products, or it could be clinical equivalence, where it's used for the exact same clinical condition or intended purpose in the exact same tissue type, in the same population, with the same type of user, and that you don't foresee any significant differences in terms of the ability to deliver a clinical performance. So really, it's assumed that to establish equivalence, you must fulfill all technical, clinical, and biological characteristics, and that differences between the device that you're evaluating and the device that you're claiming equivalence need to be identified and fully disclosed and evaluated and explained as part of that notified body review. So it's not something that you can just sort of skip through or rush. It's definitely a very exacting process. And in order for clinical data to be considered relevant, the equivalent device also should be CE marked and used in accordance with its intended purpose as documented within your IFU. So it's a very challenging process. And again, I would definitely defer to the guidance provided by MedDev in order to obtain specific information about how, if you're going about establishing equivalence, how to do that properly.
All right, thanks, Sarah. Here's our next question. This person is asking for some tips on how to assess clinical data sufficiency. What do you have to say about this one, Sarah?
So this is kind of the magic question. It's definitely a concern that we hear quite a bit. So in general, if a manufacturer is trying to decide if they have sufficient clinical evidence, you should look at whether or not the clinical data are required to draw conclusions as to the conformity of that device to those essential requirements. So the data should be in line with what's the current state of the art. They need to be scientifically sound, and they should cover all aspects of the intended purpose in all different products or settings in which that particular test may be used. So if you have a particular device that is being used in one scenario, and you have, like, let's say you have a lot of clinical data in an adult population, but within your literature searches, you're finding it's also being used in a pediatric population as well. You may have gaps in one population compared to another. So that all comes down to sort of conducting a thorough review and really assessing what types of information are out there.
Okay. Our next question is, how do you use clinical data outside of the EU to support IVDR registration?
This is something that we also hear quite a bit, this question. You can certainly use clinical data sets that are derived from studies in the US or all over the world. Global data is definitely allowed. The critical part to include here is to make sure that you're clearly documenting the source of each of the clinical data sets, and that you're still subjecting them to the same appraisal and directly explaining any potential limitations with those clinical data sets. So it's not to say that you can't or shouldn't use those global data sets if available, but the explanations and the documentation of the sourcing of the clinical data sets is going to be what the notified bodies are going to be looking for.
All right. Our next question is a good one, Sarah. I think everyone probably wants to know the answer to this. What are the biggest gotchas you have seen, and what are some tips to avoid them?
I think ultimately, especially for some of these devices, it's sort of two spectrums of the problem. Either you don't have enough data or you have too much data. And either way, it's all about telling the story. You want to make sure that you've selected the appropriate competitor devices to establish your acceptance criteria. If you're using a competitor who maybe is not a similar technology, maybe they should have been more of an alternative, or maybe you don't have a direct competitor, that can be really challenging because you may wind up not meeting the acceptance criteria that you've set if you've selected the wrong competitors. So I think it comes down to preparation, making sure that you know that you have people in place who are able to fully assess and appraise these data sets, to create the templates, to present the data in a way that's clear and concise and tells that story, and is very well organized. So that I think is ultimately in most cases when we have when we're helping a client who is trying to respond to feedback, it's often that everything is there. It's just hard for the notified body to find it. So it's often a question of reorganizing and stating what we believe to be obvious, but you have to draw these conclusions and explain how the data actually tell and meet your conclusions. That's usually what the notified body is missing. If they have to look for it, they are probably not going to. They're very much overworked and they're working under very tight time constraints. So make sure your data is very well organized.
Definitely great advice. Here's our next question. How do you support clinical data requirements without a clinical trial?
I think this also kind of goes to the point of data sufficiency. We are hearing this from a lot of clients as well. They are basically in panic mode trying to decide whether or not they actually need to have clinical trial data. So in general, if you have conducted a thorough review of all available data sets of the published literature, and there is still a gap, that is going to ultimately require data eventually. So that can be a clinical trial. It could also be a survey. So in particular, if you have a test that is not in sort of a high hazard class, a survey can be extremely low cost and easy to implement, but not quite as demanding as a full clinical trial. Now it will be appraised differently as far as the overall relevance of that data, but that could be one option that I've heard many groups suggest in terms of avoiding the need to conduct a full clinical trial. But in some cases, because this is a brand new requirement for IVDR, it may eventually be necessary to have and start proactively collecting those clinical data sets by looking at hospital databases or registries or things like that.
All right. This audience member is asking if you have any experience with a product family that has both MDR and IVDR products, and do you write a CER and PER to meet the conformity?
I would imagine that if your product family, so if I'm reading this right, if the product family would include both a medical device as well as a sort of a test. In that case, I would imagine that you would still be expected to develop a separate report for each of them. But yeah, I don't know if that's entirely clear yet as to sort of the overall scope and sort of it would really depend on sort of how the IVDR project fits into the scope of this. You can certainly reuse some of the information, but because they are different requirements, I would imagine that you're going to ultimately require both a CER and a PER. And I don't know, Lori, if you have any other thoughts on this. I'm trying to think of like an exact scenario or an example of this. So maybe it would be like almost like an implantable, I'm picturing like a glucose pump or something like that, where you could pair that to a Dexcom. Yeah, something along those lines.
I would say that the question is, do we have experience with that particular scenario? And I would say across our organization, we have not encountered that as a scenario yet that we've worked with a client on. My general thought about this is go back to the regulations under which this device, if it's a combo device, for example, or how is it on the market? Is its pathway to the market as a medical device, as a combination device, as an IVD device? Which regulations apply to it? And which is, certainly a question for your regulatory affairs colleagues. And then then you must comply to those regulations. If that's not clear, then that feels very much like a very early discussion with a notified body, your regulatory contacts and so forth, to get very specific clarity on that. But it feels like to me that at the end of the day, it has to do with the regulations under which the device was placed on the market in the EU.
Mm-hmm. Yeah, and again, kind of referring to the wearable technologies like a glucose, something that's measuring blood glucose. The device itself is implantable, so that would be the medical device. But the software that's actually recording and processing and developing that's going to ultimately send that information back to like a pump, an insulin pump, would be an IVDR product. So I would envision that you can definitely reuse some of the language and some of the background and even potentially some of the searches. But the purpose of the actual implantable device is much different than the software used to analyze the data for that implantable device. So their clinical benefits are different. So that would be the main point of distinction.
Right. So I think our advice is to have very careful conversations up front and get clarity and a decision from notified bodies, your regulatory folks, whatever it is, so that you within the organization can proceed with confidence in developing the data identifying the data that you're going to need ultimately for either a CER or a PER or even both.
Mm-hmm.
All right, thanks, Lori. Okay, here's our next question. Do all of the requested clinical performance data need to be addressed?
In our experience, in short, no. It really will depend on the IVD. I have been referring mostly to, I sort of start with the clinical guidelines, and I try to see the most common ways of assessing the accuracy and the overall clinical success of a particular test. So depending on the purpose of that test, you may be able to find information about likelihood ratios or positive predictive value or negative predictive value. In some cases, those particular objectives are not quite as commonly used depending on the field. So I think it goes back to using the guidelines, using the clinical practice guidelines and that competitive landscape to help you justify which of these clinical performance data you've identified as being the most relevant. You want to make sure that that's all done objectively and very transparently. So you don't, it doesn't appear as if you've left out a clinical performance data that doesn't support your device as being considered state of the art. You would want to make sure that you're being very objective and very clear about your choice and justify that within the PER.
Great, thanks, Sarah. We have about 15 minutes left, so if you haven't sent in your question yet, you can use the Q&A button to send us your questions. We'll try to get to as many more as we can. Here's our next one, Sarah. For legacy products, what is the scope of leveraging existing clinical trial data, which could be very old?
This, I think, again, kind of goes to the fact that many clients are scared of the prospect of having to engage in clinical trials. They really don't like the idea that they're going to have to start collecting new data. So yes, you can definitely include some of these pivotal trials, some of this older data for a legacy product, but that doesn't negate the need to do a current systematic literature review and to identify and continue to proactively collect some of that data that's currently being published. So it is perfectly fine and expected that you're going to leverage existing data, especially for those original pivotal trials. But don't forget to continue to include a plan to collect, prospectively collect that data moving forward as well.
Okay. On to our next question. What justification would work for not requiring clinical performance studies?
I think ultimately it comes down to the classification of your IVD. There are some preliminary guidances suggesting that, like, for instance, in a Class A device, that you would not necessarily be expected, or that a clinical performance study would not be expected, especially for something like a reagent or something like that. Whereas pretty much any of the other classifications, it's expected that you would be required to collect clinical performance data. So I would say that it's, I would assume that unless you fall under that sort of lowest risk class, that you should be able to identify some of these clinical performance studies within the published literature or within your own in-house data sets.
Okay, great. Our next one is a good one, Sarah. Do you have any hints on making clinical performance reporting reviewer friendly?
Yes. So templates are your best friend when it comes to starting this process and not getting off on the wrong foot. You know your data inside and out. That's your job, is to know your data. They won't be experts necessarily on your particular device. And so as you're presenting your clinical performance, making sure that you are keeping the sources of the data sets separate, that you're not mixing. I've seen this before where clients have attempted in their previous drafts of these reports to mix some of their own performance data into the competitive landscape. You can't set the bar with your own device. So in terms of the clinical performance data, being very organized, providing cross-links and cross-referencing different parts of the report to make it easier so that the notified bodies don't have to go digging for that information. If you've already presented an explanation within one part of the PER, making sure to include those links that allow them to quickly navigate throughout the entire report. But usually we err on the side of making it simpler in terms of the tabular format, particularly for these large data sets. A tabular format can be far easier to read than paragraph after paragraph after paragraph explaining individual results of studies. So we definitely like to make use of very easy to read tables, and also making sure that it's clear about any details of the study. If you can add that to the tables, that's also very, very helpful.
Okay. Our next question, Sarah, is coming from Cynthia in our audience. Follow-up question to a previous: don't you need a justification why you do not include specific clinical performance attributes in your CPR?
Yes. So for instance, let's say that you are, we've had this with some clients where, like, for instance, they say that their test doesn't necessarily have a false positive, it's a cross reactivity, or something along those lines. It did detect something that was there. So it performed as intended. It's just that in a clinical setting, that particular analyte is found in patients that don't necessarily have a particular disease. So it comes down to that explanation and that organization. So we usually include a separate section that fully describes each of the different performance indicators that we've described and why they were selected, what types of information they can provide in terms of the clinical benefit of the device and the clinical performance. And if one was left out, we would describe why, and provide an explanation as to why that particular performance objective is not really reflective of the overall clinical benefit to a given patient based on the intended purpose of that device.
Okay, great. Is there any particularity about collecting clinical data for IVD software?
Honestly, this came in our experience with software. It was a very, very similar process as what we've done with medical devices. There is far more clinical data out there with regards to software than one would expect. And it often comes down to some of the more technical aspects of the IVD software, but you can still use the software for things like positive predictive value, or the ability to make a diagnosis accurately, things like turnaround time. How well does that software, does it speed up the diagnosis process and provide a more rapid test result? So how does it analyze the data? Does it make it easier from a public health perspective to share that data set? Does it make it easier to collect that data and share it with the end user? So there are, when you're looking at the software, I think the main challenge is going to be that often the software is used in combination with a very particular platform or device. And so we are careful to capture data with regards to which device is being used to actually collect the data, as opposed to which software is being used to then analyze that data set. So as long as you are collecting the data and making sure to note any differences. If, like, let's say your software is only designed to be used in one platform, and you've identified some data sources that actually use it on a totally different scanner or a totally different collection source, that's something to make note of in your data sets.
Okay. Here's the next question, Sarah. How are search terms organized in a systematic review?
This can be very, very complex. It comes down to having a good relationship with a medical librarian, I think. When we design our systematic reviews, my role as the writer is to provide a list of the key terms as I see it. If I were trying to look up information about this particular topic or this particular test, what are some very common terminologies that I would use to find that information? But the idea behind the search strings that a librarian can craft is that they can use more advanced algorithms to ensure that you're not getting a lot of off-target hits in your searches. So they can do things like make sure that you're looking for molecular diagnostic tests within 5 words of your particular bacteria or virus that you're trying to detect. If it's a COVID test, you would want to make sure that you're trying to find a certain type of PCR COVID test, you would want to find articles that mention PCR in the context of COVID. If it's one or the other, you could have many, many, many hits. So it's definitely an iterative process, but the documentation of this and the ability to work with an experienced librarian who can help you with properly crafting that terminology is very critical to not having a massive number of articles to sift through, because you will ultimately have to assess and screen all of those articles, and you could have quite a few of irrelevant articles that you will then have to document in the process. So it's definitely worth your time to bring in a reference librarian. I think on average, for the average company, they estimate that this process of conducting the systematic literature review takes a minimum of 8 weeks. And that's a fairly solid estimate if you know what you're doing, I would say. So it's a long process. It's a very time-consuming process, and it definitely requires an expertise.
Definitely. And if you're looking for more information about medical librarian services or how you can obtain these services for you or your team, reach out to us at consult@Criterion Edge. We'd be happy to set up a quick call with you and give you some more information. Here's our next question, Sarah. Do all devices require a clinical performance report, or does this only apply to certain device classes?
Yes, I think that gets back to sort of the class A versus sort of the higher classes. It's generally acknowledged based on the current guidance that we do have that a class A would not require a CPR, a clinical performance report. You would still explain why within your PER, but for the other classes, the CPR is a requirement. So, and that's a key distinction between IVDD and IVDR is the drastically expanded requirements for clinical performance data.
Okay, we have just a few minutes left here. I'll get to a few more questions. Here's our next one. What are acceptable sources for clinical data? What can you say about this, Sarah?
I think you can definitely start with your systematic literature review and see what information's published out there. It kind of comes down to internal versus external. If you're lucky, you have that internal data sets, you have manufacturer held data, but you're often relying on the external data sets, and those are where having the ability to search across multiple platforms and databases is really critical. It can help you identify articles that have published clinical data, and ultimately having a properly designed search can save you from having to conduct your own clinical trial in some cases if you're just not searching properly and not finding the clinical data sets that are out there. You know, you may be able to identify registries or identify databases where this information is already available to you, or you may be able to conduct surveys to help you support some of the existing clinical data. Ultimately, if there is just no clinical data available, that's where it may be necessary to plan either a survey or plan a study internally to be able to show that you're proactively and prospectively collecting this data in the future.
Okay, great advice. Here's our next question, and maybe a big one, Sarah, but do you have any tips for IVD data collection?
Organization is critical. Start with the guidelines. See if there are any guidelines specifically pertaining to the accuracy of testing. Identify who your alternative ways of making that clinical diagnosis is. If your test is not available to a person, how would they make that same diagnosis? And then being accurately identifying your competitors, finding competitor devices that have the most similar characteristics to your device. Often that information can be found by working with your marketing department. They often know who your direct marketing competitors are. But when it comes to data, it's then working with the reference librarians to craft searches where you can actually find the data that's unique to your competitors, and to be able to identify the correct performance measures that are supported by those clinical guidelines. So it comes down to having a process in place to where you know what information is available to you, you know the current state of clinical practice guidelines, and you're aware of who your competitive landscape is, so that makes the overall process easier to justify in your PER.
Okay, great. We have one last question, Sarah, here for you today. How is the benefit risk profile evaluated from the clinical data?
In that case, it's going to ultimately tie back to your acceptance criteria. You've established what can be considered acceptable performance in a clinical setting. If you are trying to, your role in the benefit risk profile then is to use that acceptance criteria to show how your device compares to your closest competitors, whether or not it achieves the same sensitivity, specificity, rates of false positives or false negatives. And if there is any scenario in which it does not meet the level or the bar that's been set by that acceptance criteria, being prepared to explain why that's an acceptable benefit risk ratio. Your device may not be the most sensitive available, but if it's a point of care device and it can be used in resource poor settings, that's a benefit. So it comes down to making the case, but also showing that the bar that you've set was set objectively, and that you are also collecting your data very objectively, and that you've done a thorough assessment of all of the clinical data that's out there.
Okay, we are out of time. Thank you so much, Sarah, for your presentation today, and everyone for your questions. For more questions on the content we discussed today, or if you'd like to learn more about our services, you can schedule a free appointment with us for attending. Our contact information is consult@criterionedge.com, or you can book a meeting time directly with the link in the chat, which my colleague will share with you. Thank you everyone for attending today and for your wonderful questions. Thank you, Sarah. We hope this presentation helps you get one step closer to IVDR readiness. Take care, and I hope to see you at our next webinar.